Ciao ragazzi! This is our second and last section of the immune system. So we are now doing adaptive immunity. In fact, actually, I should run up and look at this review. So remember, we covered last time innate, non-specific immune system or immune response, which included all this stuff, right? The wall and the second line of defense, phagocytosis, inflammation, the fever. And we are now going to cover specific immunity or adaptive immunity, which involves the lymphocytes and antibodies. Okay, so that's where we're at. To review, I recommend watching this video just up to 3 minutes. It's 3 lousy minutes. I know you can find the time. It'll be a nice review of what we've learned. And let's begin the new material. Ta-da! We start by labeling the bad guys. What is the name of the bad guys? The bad guys are called? Yes, pathogens. That's correct. The bad guys are called pathogens. And the bad guys have lots of surface proteins, just like all our cells do. Remember at the beginning of the year, we spent a whole chapter studying membrane proteins. There's lots of proteins on the surface of things. And these surface proteins have a very specific name. They're called antigens. And they're very important because it's the way that our body identifies these things as foreign, as unknown. Remember that our surface proteins on our cells help our body to identify the cell as belonging to us. My surface proteins on my cells say, belong to Mrs. Ratti, good guy, part of your body, don't attack it. And these instead say, foreigner, invader, should be attacked. Okay? All right. Now, the famous protein we're going to talk about, the famous thing we're going to talk about is antibodies. Antibodies are not cells, they are proteins. And they're proteins with this funny little shape. They're shaped just like a Y. And that becomes very important, as you'll see later. So there are proteins that are made by lymphocytes. They're Y-shaped. And on each tip of the top of the Y, each of this double tip, there is something called, there is something called an antigen receptor or an antigen binding site. It's the same thing, an antigen receptor or an antigen binding site. So this here and this here are antigen binding sites. Right here in orange is another set of binding sites. This is another diagram. You can see in this diagram, they made the antibodies with brightly colored binding sites. Here they're green, here they're light blue, and so on. These binding sites or receptors are specific. They're made to bind with a specific bad guy, specific pathogen. So this one here is made to bind with this bad guy, with this pathogen. This purple one here is made to bind with this bad guy. Here they don't show the pathogen or antigen, but you can tell that they're going to specifically bind with something in particular. That's why they're made in different colors to represent that. Okay, so just to make sure you get all the terminology down, can you label each of these? 1, 2, 3, 4, 5. Label each of those. Take a moment to do that. And ta-da! I put all the bad guys in red so you wouldn't get confused. Now, to be precise, the bad guy is called the pathogen. The antigen is its surface protein. But to be honest, when speaking of immunology, lots and lots of times, textbooks, articles, doctors, they're vague. They kind of refer to an antigen or a pathogen in the same way. Because once you've hooked on to an antigen, you've hooked on to a pathogen. So sometimes you need to be precise. Sometimes they're sort of interchangeable. Don't let that worry you. Okay. Antibody is meant to refer to this whole thing, right? The entire antibody. Okay. So again, just to identify it, if this purple guy is a bacteria, the purple guy would be the pathogen. The green surface proteins would be the antigens. Don't worry about this label. It's being more specific than we need. These would be the antigens. The red guys would be antibodies. And this little part of the red guy here that's attaching would be the antigen receptor or the antigen binding site. All right. So as in these pictures, there's antibodies that are found floating around loose. But many antibodies and the origin of antibodies are found on the surface of B cells. To be more precise, they're called B lymphocytes. So let's look at that. This is a B cell. To be precise, it's a B lymphocyte. And this B lymphocyte, what has it got on its surface? Its surface proteins are, you recognize the Y shape, they're antibodies. They're antibodies. Here they're calling them membrane-bound antibodies, meaning they're just bound, tied to the membrane of a B cell, of a B lymphocyte. different B cells are coated with very different types of antibodies. So this picture represents it easily. There's a triangle shape, a square shape, a circular shape. These all represent different types of antibodies. Why? The antibodies are specific to different pathogens. This can bind to one type of pathogen, this will bind to another, this will bind to another. So these are B cells specific for different antigens slash pathogens, okay? So B cells are different because they have different B cells which are specific for binding to different kinds of pathogens slash antigens. Okay, this picture here, each type of B lymphocyte will bind to its specific corresponding pathogen. So this B lymphocyte here, it's got antibodies, and its antibodies have binding sites. But can it bind to this pathogen, to this illness? No, it cannot. Can it bind to this pathogen? No, absolutely not. They're all specific. Only this guy can bind to this pathogen. Only this guy with the red antibodies can bind to this red antibody. And this guy will have some other pathogen, which is not present in the picture. It's not present in this person's body at the time, supposedly, but it will have some other type of pathogen it can bind to. Obviously, the imagery of colors represents something much more complex. It's a specificity that's chemical, right? But you get the idea. Using colors and here using shapes is a good way to represent that chemical specificity. Okay, so, this picture is just a little bit more detail, so let's just look at it. This entire thing is the pathogen. So this is some big, terrible bacteria or virus. It's full of many surface proteins. These are kind of cool-looking surface proteins, which of course we know are called antigens. And here's a B cell coated with, what is it coated with? Many antibodies. Each antibody has an antigen receptor, has 2 antigen receptors. And look what these antigen receptors can do. They can bind. Here it shows that specifically it's binding to the antigen, but obviously when it's bound to the antigen, it is now bound to the entire pathogen. That's why there's an ambiguity in the term. And it's specific. This guy cannot bind with many, many, many pathogens. It can bind with this pathogen. Okay, so the first step in the immune response here, a B cell's antibodies bind to a pathogen. It finds a pathogen that it can bind to and binds. To be more specific, when it's binding to a pathogen, it's actually binding to the antigens on the pathogen's surface, okay? And what happens at this moment, right in this moment right here, what you're seeing is the B cell is activating. It's waking up. It's actualizing its superpowers, okay? It's undergoing a transformation. It's like Superman spinning around, changing out of his Clark Gable glasses and goofy suit, and instead getting on his Superman suit. It's happening right here, activation. And it's only this B cell is being activated. Other B cells that can't bind to this are not being activated because it's this act of binding which is going to activate it. That's actually simplification, but it's close enough to the truth. Okay, so I don't know why I have this picture again. You can look at it again. It's a useful picture, but let's move on. So in this picture, which B cell or B cells are being activated? The answer is pretty simple. This guy's getting activated. This guy's not. And what do you think? Look at this binding going on. This guy's getting activated. So here we have 2 activated B cells. Now let's look in detail what happens when a B cell is activated. Oh, wait. First, okay, another image. I know there's lots of images here. These all represent B cells with lots of antibodies on their surface. This is B cell with gray antibody, B cell with blue antibody, and let's call this B cell with turquoise antibody. Okay, now a bad guy has entered the body. An antigen slash pathogen has come into the body and it's floating all around. It got into your body fluids or maybe even your blood. It's floating around and look what's happening. There's a B cell that found that it can bind to this antigen. Can it bind to these? No. Can it bind to these? No, the binding site doesn't match up to that antigen. It binds to these. So which one is getting activated? Ta-da! This is the activation, the one being activated. This is the one getting on its red cape. Okay, so now it becomes a little bit of a complicated process, but it actually, if you understand it step by step, it really makes sense. Once it's been activated, so here's the picture we just looked at. This guy has been activated. What does he do? He proliferates. That means he reproduces himself over and over and over and over, right? He makes more life. He makes more of himself. We could say he clones himself. Okay, he reproduces himself many times. Here we're just showing 2, but it's going to happen to many copies of these. Each copy makes more copies, okay? So they keep cloning themselves and cloning themselves and cloning themselves until there are tons of identical cells to this original B cell. Now, I know these cells look different, but there's really only two reasons why they look different. Okay, you ready for this? This is important. They look different because what have they done with their antibodies? They have secreted them. They've secreted them into your blood and your lymph. They are sending these antibodies all over your body to go get the bad guys. Okay, so they've secreted your antibody. The other reason they look different is they look a little bit bigger and longer. This stuff represents endoplasmic reticulum, rough endoplasmic reticulum, because rough endoplasmic reticulum is the stuff that makes proteins. Antibodies are proteins. So why do these have lots of rough ER? Because they're going to make tons and tons and tons and tons and tons and tons of antibodies, and they're just going to basically become antibody factories, producing a million bajillion antibodies and secreting them into your body fluids so that your body has literally millions of antibodies floating around. Okay? So these look different from this guy, but they're not. They're exact clones, just with more rough ER and with their surface antibodies having been secreted. That's the only difference. Okay. So let me just read the summary. Once a B cell is activated, it clones itself, produces many copies of itself. Each copy is an antibody factory. The purpose of this cell's life is to produce many, many, many antibodies. Okay, and this explains what I just explained a moment ago. So the name of the copies or the clones are called plasma cells. And this again explains what I talked about earlier. So read it again. Just to review it, what the purpose of the rough ER is, rough endoplasmic reticulum. Now, what is the good of antibodies? Why are they so important? What do they do? You know that they bind to antigens, but what good does that do? So here's a picture of a pathogen. Here's our red pathogen. Its black dots are its antigens. And here is a picture of antibodies which happen to bind to that. Now again, the fact that antibodies are Y-shaped is really important, that they have two binding sites is really important. Each antibody is going to bind to two different sites, right? And because of that, what happens is it starts to join together. I use the word clumping a lot, to clump together a lot of the pathogens. Because there's two sites, this antibody is connecting this one to that one, this one's connecting that one to that one, and so on. So they start to clump together. So let's read this. When many antibodies are released and they bind both of their sites, both sites of the Y, to antigens, then clumps of pathogens begin to form. This has a fancy name. Are you ready? It's called, there's just this clumping, what we're naming. It's called agglutination. And it's really easy to remember. This is a silly association, but agglutination sounds like glue because it gets gluey or clumpy. Okay. Here's another better picture of agglutination. It's the clumping of antibodies and pathogens. And once these antibodies are gathered up, they become neutralized. That means they're like immobilized. They're like they've got handcuffs on. They can't act. And also, they're easily found by macrophages and other phagocytes, and they get eaten up and destroyed. The macrophage has no problem finding this huge mass, this huge agglutination. So it chomps it up and destroys it. Whereas a tiny bacteria or virus can slip away from a macrophage and can attack. Okay, another picture of the same thing. This is neutralization. It's surrounded by antibodies, this virus and this bacteria. It can now not attack a human cell. It's been neutralized. Without its surface proteins, it can't enter and attack a human cell. Here, it's been agglutinated into a clump. Basic same idea here. Okay, so here's one last super important, really interesting detail. You recognize this picture. We've studied this so far. Right? Here's the clones, which are antibody factories, but there's one which remains for a long, long time. It's called a memory cell. After a sickness is over, your body keeps some of those B cells with a specific antibody for fighting that specific sickness. It keeps them or stores them in lymph nodes. Lymph nodes spread throughout your body, also in your spleen, in case that same pathogen ever returns, in case you ever encounter that same disease. These memory cells can be kept for many, many years. And if the pathogen ever returns, the immune response will be fast. So fast that it'll be like not getting the sickness. You'll get very few or no symptoms. Oh, I'm out of time.